Hi everyone, my name is Shomnak Banerjee. I am a research scholar in Bose Institute, Kolkata, and today I will teach you about nerve impulse transmission through a neuron. So what is the primary function of a neuron? The primary function of a neuron is to transmit the nerve impulse from a previous neuron to a postsynaptic neuron. So how does that happen? The first step of a nerve impulse transmission comes from the neurotransmitters which are secreted by the previous neuron. Now these neurotransmitters act as a chemical messenger that when binds with the dendrites of this neuron, it binds with some ligand-gated channels. Now what are ligand-gated channels? Ligand-gated ion channels are some ion channels which will open in response to binding of some ligands, and these neurotransmitters are those ligands. Now, what happens when the neurotransmitter binds with those ligands? One thing is to be said previously, before I come to this, is what is called resting potential. Resting potential is a potential difference, you can say, between inside the cell and outside the cell. It is achieved via maintaining concentration of sodium and potassium and other positive and negatively charged atoms or ions inside the cell and outside the cell. Now, how is that achieved? That is primarily achieved with the help of some ion channels, some ATPases, the main of which is sodium potassium ATPase. Sodium potassium ATPase functions by hydrolyzing ATP, and it hydrolyzes ATP to ATP and PI, and the energy it gets from hydrolyzing ATP it uses to transport 3 sodium ions from inside the cell to outside the cell and 2 potassium ions from outside the cell to inside the cells. If you note carefully, there are 3 sodium ions migrating from inside the cell to outside the cell and 2 potassium ions migrating from outside the cell to inside the cell. So there is one positive charge migrating outside the cell. That is responsible for formation of this inside negative resting potential. The normal resting potential for a neuron is generally minus 70 millivolts. So that is called inside negative, minus 70 millivolts inside negative. So when a neurotransmitter secreted from a previous neuron binds with the dendrite of this neuron, some ligand-gated channels open. As we know that there is a scarcity of positively charged ions inside the cell, and there is a flooding of positively charged ions outside the cell. So it will move, the positively charged ions will move across the concentration gradient from outside the cell to inside the cell. This will bring up the potential, the membrane potential, from its resting potential, which is minus 70 millivolts to minus 55 millivolts. Now there are very other ion channels which are present in the axon or in elsewhere in the neuron. Those are called voltage gated ion channels, meaning that they will open only in response to certain voltages, and minus 55 millivolts is that particular voltage. So when the cell or the membrane potential attains minus 55 millivolts, all the voltage gated ion channels open. As a result, all the sodium ions which are present in more concentration outside the cell compared to inside rushes in. So sodium rushes in. As there is a common knowledge that when there is a inside negative membrane potential and a positively charged ion rushes in, this will naturally result in depolarizing the cell, that is moving the membrane potential more towards positive. Okay, so this becomes a reality when sodium ions bombard in or rush in until the cell or the membrane reaches a potential of plus 40 millivolts. Can you imagine how polarized it is? Plus 40 millivolts. When it reaches plus 40 millivolts, so minus 70 millivolt is called resting potential, which is the potential of the membrane when the cell is resting, and minus 55 millivolts is called threshold potential, a threshold when all the voltage gated sodium channels will open. So when the cells on the membrane potential reaches plus 40 millivolts, then the sodium channels close. Okay, in response to that voltage, the sodium channels close. So the membrane, so there is no ingress of sodium channels now, and through the leaky channels the sodium goes out, or via the sodium potassium ATPase also the sodium is starting going out. So the membrane is negative now. It goes towards negative. So the impulse from here moves. So this becomes positive. Now this, in response to this positive charge, the voltage gated channels of the next node opens. So this becomes positive, and this starts this negative phase, which is known as the repolarization phase, or the it is called it is known as the refractive period, when the nerve cannot fire. So in this phase, the sodium channels open, the potassium channels open. So as we know that the potassium concentration is inside higher and outside lower, so the potassium rushes out. Okay, when the potassium rushes out, this drive towards more negative membrane potential fastens. So this is more fast, and it reaches also below the resting potential. Okay, this is when the sodium channels shut off, and subsequently slowly this through the other physiological conditions and leaky channels, the membrane potential rises up to its normal resting potential. Now the neuron can fire. So what is the significance of nodes of Ranvier now? So as we know that the axon of majority of neurons are covered with myelin sheath. So there are very little, so myelin sheath acts actually as an insulator. So where all the ion channels, the voltage gated ion channels and the ligand gated ion channels are covered. So this prevents leakage of ions from inside the cell to outside, and also it speeds up the conduction of nerve impulse. How? There are small gaps between myelin sheath covered areas which are known as nodes of Ranvier. So only at the nodes of Ranvier, the sodium and potassium channels, which are either the ligand gated ion channels or the voltage gated ion channels, are exposed. So only nerve impulse the sodium potassium exchange from inside the cell to outside the cell can happen only through these nodes. So the positive charge actually jumps from a node to another node, thus fastening speeding up the process. So when the positive charge reaches the axon terminal, then what happens? Then there are some vesicles which contains neurotransmitters, as the neurotransmitters spoken about at the first of this class. These vesicles merge with the synaptic membrane, and subsequently there is a release of neurotransmitter between the junction between this neuron and the postsynaptic neuron. Subsequently, these neurotransmitters bind with the ligand gated channels of the postsynaptic neuron and starting the process again. So this was the class about nerve impulse transmission. Hope you like it. If you like it, please give a thumbs up.